Hello, plant propagators. Welcome to this online lecture. Thanks for your patience this week as I am off to a conference. I will be available via email. I might not get back to you right away because the conference is all day. But please send me emails if you have questions about this lecture or any of the content from our first few weeks here. We're ending our unit on seeds. Your exam is this week. Exam number 1 is this week. It opens up on Wednesday and will be available through Sunday evening. So please make sure to submit that exam and you won't lose any points there. To prepare for that exam, there is a lunchtime study session on Wednesday at 11:30 in the main floor classroom. So that classroom that's off to the left as you walk into the door. And our amazing tutor, Emily, will be there guiding you guys through a study session. Please attend that. Please go, bring your lunch, ask questions, and talk about the content with Emily there. She'll give you guys some study tips as well from her experience. Emily's a senior. She's been here for a few years now and has a lot of wisdom to share about how to prepare for exams here in the CWI horticulture program. So this would be a really good opportunity for you to learn from a peer and support your fellow classmates and study up for this exam. For the exam, you can use your notes. You can use anything on Blackboard, including our readings, things like that will be included in the exam. There's 50 questions, so please set aside enough time to do the exam. And always reach out to me via email if you have any trouble accessing things, questions, etc. So without further ado, I'm going to launch into this lecture here about plant selection, and we'll learn what that is and why it's important. We're going to back up and we're going to look at how plants are named. And how plants are named is based on their taxonomy. So taxonomy is just the classification of living organisms. You may have seen this chart before, maybe in a previous biology class. Kingdom, phylus, class, order, family, genus, species. Should look a little bit familiar, but we're really dealing with these last 3 primarily here in the family, genus, and species. But I wanted to show you kind of how we get from point A to point B when we're naming organisms, so naming plants or naming insects or naming fungi or whatever it is that we're looking at, us, for example. And actually, we're going to go ahead and use Dr. Evil as our example. Dr. Evil is the bad guy from Austin Powers. If you haven't seen it, it's a goofy series of movies, and you'll see why we're using him in a second. So Dr. Evil is an animal, right? As are you and I. So we're in the kingdom Animalia. We are in phylum Chordata, which just means we have a spine. Invertebrates, or things that don't have vertebrae or a spine, would be in a different phylum. So they're in maybe they're in Arthropoda, which is like the insect phylum, or they're in another phylum like jellyfish or something like that. You can keep going more and more specific. So you could be in the class Mammalia, which makes you a mammal, right? Order Primata. So, and we're all in the order Primata, meaning we're primates, right? Relatives of monkeys and other things. We're hominids, or in the family Hominidae. That means that we stand up on 2 legs and kind of do a lot of things with our hands. We're in the genus Homo and species Sapiens, right? So that's human beings for you. Neanderthals and other like related species would be in different species. I don't know which ones those are, but they're in Homo and then they're in other species. When you look at Dr. Evil, or if you've seen this series very much, he has a clone of himself that he's made to be meaner, angrier, and shorter than him. So we would call him maybe a cultivar or a cultivated variety of Dr. Evil. So we'll call him Mini-Me. Or he is called Mini-Me. So let's apply this principle to a plant. So we're going to use Japanese maple. You can see this nice leaf here on the right-hand side of the screen. We'll use Japanese maple. Japanese maple would be the common name that we would use. let's find out what the rest of its name is comprised of. So Japanese maples are in the family Aceraceae, which is the maple family. And you'll learn more about that when you get into trees and shrubs. Families, we write capitalized, and we have this AE on the end. So if you have a family name, you always need to have it capitalized. You don't do anything else to it, just that capitalization. Genus is also capitalized, but it needs to be italicized. So our genus here is Acer, italicized, capitalized. It's really important to follow these naming conventions so that you know kind of what term that you're using. It's a way that we can communicate regardless of language. The binomial nomenclature system, which just refers to this genus and species, is used worldwide to talk about plants. So you can label things in really any language, and it's always going to be the same. It's always going to have that Acer genus, which is pretty cool that we've come up with this. So species usually follows genus, and it'll be uncapitalized and italicized. So that binomial name is the genus, it's the species, they're both italicized. For our friend, the Japanese maple, that's going to be Acer palmatum. So palmatum is the species. And you can kind of see that it's named palmatum for the palm-like leaves. So we have variety that some species can have. We've talked a little bit about variety before. It's basically a naturally occurring difference in some sort of feature. So could be shorter. It could be that it's got a purple flower instead of a white flower, or vice versa. It could have a yellower leaf. You name it, right? But it's a naturally occurring difference that was found, not something that was intentionally cultivated. So we write that by it's going to be uncapitalized. It's going to be italicized. Oh, I'm so sorry. This dissectum should be italicized. I will change that in our slide notes. Shame on me. Should be italicized. But you do write this VAR period in front of the variety name. So it goes VAR, which stands for variety. And then dissectum is our variety we're using here, which refers to its branching habit that this particular variety of Japanese maple has. So you may be asking where cultivar comes in. Well, you tag that on at the very end. The very last thing is the cultivar. Cultivar is capitalized, and you use single quotation marks. So both terms, if there's multiple words, they're all going to be capitalized, and they'll be surrounded by single quotation marks. And again, cultivar, or cultivated variety, is something that was like intentionally bred to be a certain way. So for this example here, Crimson Queen is a cultivar that was generated, I think, in the 1960s, and was built to have really, really lovely fall color. And here is a picture of that. So this is our name that we've got here. Finally, I've got dissectum right. So Japanese maple is our common name, and then we have Acer palmatum variety dissectum Crimson Queen, and she looks like this. So gorgeous and beautiful. Now, one thing that is important when you're referring to plants just in like the middle of a sentence, if you're referring to a maple, for instance, you don't actually capitalize that maple. So you can see here in my common name up here, maple is not capitalized. Japanese is capitalized because any like proper nouns will be capitalized. So if it's Norway maple, the Norway is going to be capitalized, or if it's somebody's name, that will also be capitalized. So proper nouns are capitalized, everything else uncapitalized unless it starts at the beginning of a sentence. So those naming conventions are really important, not only for this class and all your other classes here at CWI at horticulture, but for industry standards in general. So when we're looking at plant breeding, this term that you may have heard of before, we've talked a little bit maybe about, we're using our knowledge of things like genus and species to create new cultivars, new find and discover new naturally occurring varieties. And we're doing this to make lasting heritable changes. So things that can be passed on from one generation to the next, right? It's not just like a fluke and it goes away. And these are intentional changes that will usually better our lives somehow. So this is, we're very human centric here in plant propagation. We're doing things for human purposes, and we may be breeding plants for human benefits. Some different examples of that could be in enhancing the plant's performance. Performance is a big term that covers lots of different things, but it usually is the plant's response to stress and how well it does under certain stressors, or how efficient it is at using water or things like that. So in this example here, we have a drought tolerant wheat here on the right and a not so drought tolerant wheat here on the left. So they were given the same amount of water. One variety is doing great, lots of green growth and leaves. One variety is starting to brown out and have some problems. So that would be a difference in performance. We could also improve disease resistance. So this example, Emerald Beauty Cotton Cotoneaster, is actually a cultivar that my ornamental plant breeder teacher at Oregon State created and patented, which we'll be talking about later in the semester, plant patents and why that's important. So he created this new cultivar, which is more compact, but it's also has improved resistance, so more resistance to a disease called fire blight, which is a bacterial disease that's really difficult, especially there in Oregon and other parts of the Pacific Northwest. And so this particular cultivar that he patented and created, and he spent years and years breeding this out in the field and in the lab, and created this new plant that is able to resist this disease. You might breed a plant to have better nutrient value, or more flavor, or a specific type of acid flavor, or something like that. You might breed a plant to be a specific color. Maybe you want this color flower, or this color leaf, or this color needle if it's a conifer, right? Or the shape. That's a big one that people will do. Ornamental plant breeding will be like, this one's smaller than this cultivar, or it's more columnar, or it's much taller, right? So there's a lot of different things we can do when we're plant breeding, a lot of different goals we can have. Mortgage lifter tomato is an example of this, that was bred way back in the day to have just an enormous tomato. So mortgage lifter, so it's supposed to lift, I guess, lift your mortgage, or like get rid of your mortgage because you have so many tomatoes that you are swimming in crops, right? Swimming in yield, I guess. So we have these massive tomatoes. And that's one example of a goal you could have in plant breeding. You could also have your breed plants specifically for livestock, so you can make the plants more digestible, or have some other value for livestock, and there may be more nutrient value for livestock, that sort of thing. So there's a super broad range of reasons why we breed plants, and how do we do that? Well, one thing that we can look at when we're breeding plants is there what's called seedling vigor, or seed vigor. So you have one example here on the left. This is a seed lot that has low vigor. So you can tell it's not very uniform. It looks like some of them are slow to germinate, if they're germinating at all. So their density is more random. And then this plant on the left here has a higher vigor. So we have uniform emergence, it's much denser, that sort of thing. And this is something that we can do when we're looking at plant breeding. This could be a goal of a plant breeder, to have a seed with like a higher vigor, which that vigor just kind of means like the strength of the condition of the like the genes inside the seed. So vigor is like how well that thing is it's related to that term performance we talked about. How strong is it? How lush is that growth? How dense is that foliage, or how dense are the seedlings when they come up, right? And that's kind of different terms for vigor that we use. And when we're looking at something that has high vigor, it may perform well under a wider variety of circumstances. So let's say that we don't want to use bottom heat for some reason. A seed with high vigor is going to be more adapted to that wider range of conditions: no bottom heat, less water, changing, fluctuating temperatures, that sort of thing, than a seed with low vigor that like really needs all of those precise, right environmental conditions in order to have that uniform emergence and performance. So when we're looking at plant breeding, how is this actually done? Well, we have two main terms here. We have crossing or interbreeding, and we have plant selection, or a lot of times we'll hear about like roguing. So crossing is where you're deliberately taking pollen from this plant and making sure it gets to pollen on that plant. And there's a lot of different ways to do that. You can take a pollen brush from this plant to that plant, right? Make sure that no pollinators are doing this, you're doing it, you're hand pollinating, right? Or you can quarantine plants in a certain area and have a lot of space between another group of plants that they might accidentally get pollinated from, and make sure that they're like separated from another population, so that they're only breeding within themselves, and that would be like crossing all those genetics in this one area. So we given that example a lot of like this cherry up here on this mountain top and this cherry up here on this mountain top, and they're physically separated, which means they're interbreeding here and here but not between each other. And so when you're a plant breeder, there actually are very specific distances that you can grow certain plants away from one another to make sure that those plants are separated, so that you're only crossing these tomato plants or these corn plants, not your neighbor's corn plants, and corn plants that are nearby. So that density is really dependent on the plant and how it is pollinated, whether it's wind pollinated or pollinated by insects. Insects travel a lot longer of a ways. Wind pollination usually happens pretty close by, surprisingly. So when you're working with crossing plants, you're interbreeding plants that are closely related. So either in the same genus, or maybe they're the same species, but they always have to be the same genus. So it's very unlikely that you'd have two different genuses. So like a maple and a spruce, which are two different genuses, they're not going to cross pollinate each other, right? But two maples next to each other might cross pollinate, or especially like two maples that are in the same species. So two Japanese maples are way more likely to cross pollinate. So it gets more and more specific as you get closer to that like species level. Plant selection is another tool we have in our toolbox. We want to do plant breeding. So you're propagating plants with very specific characteristics, and then you're removing the ones that don't match. So this an example of this that Daniel and I are doing is, so we are cross-pollinating wild lettuce with some different cultivars of lettuce that we've used. So we'll grow out, like the last couple of years, so we've been growing out these like 10 by 10 sections of lettuce in a couple different parts of our yard, and we'll do taste tests. So if it's like, hey, that is way too bitter, it has way too much of like the wild crop relative or wild cousin bitterness to it, we'll go ahead and just remove that plant before it starts to flower and it's cross-pollinating with the other plants around it, right? So that's called roguing. So you're removing something that you don't like For some reason, looks like this example here looks like maybe soybeans, and he's got some plants here that there's like a little bit of yellowing going on, so maybe they were less drought tolerant, or whatever qualities he's looking for specifically. He's taking out the ones that don't match those qualities. We also have these terms cross-pollination and self-pollination. So when we're looking at how pollen is transferred on plants, are they doing it between plants? So this would be like between plants of the same different genotypes. So that means they are not exact clones of one another. They have slightly different genes. So we'll have one flower over here. These could be the same species, right? Just like you and I are the same species. We're both Homo sapiens, but we have different genes. We're not clones, right? So we have different genotypes, you and I. So we have pollen from one plant over here transferring over, usually by our bee friend, over to another plant, going down and making seeds, right? So this cross-pollination is between plants of two different genotypes, not between clones, whereas self-pollination is an example of where a plant can pollinate itself. So they actually don't need anybody else. They have all the genes right there. They are still transferring pollen. So this is still considered sexual reproduction, even though it's a clone pollinating itself. It's wild. Tomato plants will actually sometimes do this. There are a number of other plants that will do this. But yeah, they're just transferring pollen from one part of itself to another. There are some extreme examples of this where there are plants that create these flowers that don't even open to the outside world. So there's no possibility that any outside pollen is actually going to get in and accidentally pollinate it. It like wants to be self-fertilized or self-pollinated, right? So this one example here that's pretty fascinating is viola, where we see viola or violets like popping up. We see those flowers, and pollinators do too. And so there's gene transfer, happening with pollen and that sort of thing. But they create a different type of flower. They create a totally separate type of flower that exists underground, basically, like right at the ground level here. And those are called cleistogamous flowers. So cleistogamous means like closed marriage. So cleistogamous means they're not marrying anybody else. They're marrying themselves, and they are just fertilizing their own ovules. They're pollinating their own stigma. There's no genetic transfer coming in. They're just using their own genes to create seeds, which is wild. This also happens in peanuts, and notably, I just learned this, cheatgrass does this too, which is crazy. So all plants that do this are going to have some version where they can get new genes in, right? They're at some point there's going to be new genes that are introduced. They're not just going to be inbreeding with themselves forever, but it does mean that their traits will stay really, really consistent for a very long time. So I'm interested in this with cheatgrass and what that might mean for cheatgrass populations. Are they more susceptible to problems because of all that inbreeding, or is this actually like a really beneficial strategy that they're basically like using themselves to pollinate over and over again? It's fascinating stuff. There are on the extreme other end of things, there are actually plants that can't do that at all, that like if their own pollen lands on their stigma, they don't make a seed. Like that pollen doesn't germinate and go down the the pollen tube and go down into the ovule. Something in the system goes, nope, not gonna happen for me. I'm not gonna pollinate myself, and I can only accept pollen from like a close relative, the same species, that sort of thing, from a different plant that's got a different genotype. So there are a number of plants that will do this, including lily and cabbage, petunia, almond, apple, cherry, and plum. Interesting stuff. One note here, when you see things written like this, like apple, malus, and cherry and plum, prunus, versus like almond over here has prunus dulcis, so we have the genus and the species. What this usually means is that you have like multiple species in that this is being applied to. So like cherry and plum, there's multiple species of that. Apple, there's multiple species of apple, right? So it's not all just Malus domestica, which is the one that we use. There's also like other wild type apples out there. So just a note, when you start to see these things, you'll start to understand how and why things are written. So let's look at some different categories of cultivars, because you may be thinking in the back of your head, like, okay, plant breeding, like what can I save seeds from in my garden, and what maybe can't I save some seeds off of? And you may have heard this term before, open pollinated. So open pollinated cultivars are just those that produce seeds that are true to type. So they will look exactly like, or very, very close, share the same characteristics of the parent plant that were like important to begin with. So this example here of a brandywine tomato, if you save seeds from a brandywine tomato and you grow those, you plant those seeds and you grow them out, they're going to look like a brandywine tomato. Heirloom is another term that you'll hear a lot of the time. And that just means it's a cultivar that's older. It's just a cultivar that's been around for a long time. Most of those are open pollinated. At some point, that term may apply to some other cultivars as time goes on. But right now, this just refers to cultivars that were bred or developed more than 50 years ago. So they've withstood the test of time. People still like them, or some of them. And that means, they may have some greater value in that way. A lot of that's very subjective and up to like the consumer, the market, the grower, whether or not things are better than the other. It really depends on the system that you're using, and the scale that you're using things at as well. Then we have this term F1 hybrid. There's a lot of specifics to this. We're just gonna talk about it in terms of what's true to type and what's not true to type. F1 hybrids are not true to type, will not be true to type if you save those seeds. So if I save seeds off of this example here, which is celebrity, if I save those seeds and I grow them out, they're going to look all sorts of different ways. So they will look, some of them will be like shorter, taller, bigger. Some of them may not even have red tomatoes, right? They may taste terrible, and they're not going to have the same genetics as the parent plant. So if you save seeds off of celebrity tomato or any other F1 hybrid, those seedlings that grow up are not going to be true to type. And what this really means here, so this is a hybrid, so that's a planned cross between two different genotypes. That's what a hybrid is. So two different genotypes, smash them together. There you go, that's a hybrid. It's in this example here, both of the parent plants of our celebrity tomato were self-pollinated for a really long time. So both of my two parent plants were like self-pollinated for a really long time, which means those genes that like produce those certain traits that we liked were like really, really stable, because we didn't have any other genes like coming in for long periods of time for many generations upon generations between like this parent plant and this parent plant. And then those two like inbred parent plants got crossed together, and that is our F1. So that's our like first generation, is what that stands for. So F1, first generation of a planned cross between two pretty inbred plants. And when you do that, when you cross those two inbred plants, the seeds of like that F1 hybrid, so that first generation, are just not going to be true to type because you've done some kind of wacky things to the genetics. So let's look at an intentional cross here that was done many years ago. So you may recognize this maple over here, or at least have maybe undoubtedly you have seen this tree. This is the Starbucks of trees nowadays. It's on almost every corner. If you go anywhere in Eagle, it's there. So Acer freemanii, one of the common cultivars is Autumn Blaze. And how did we get to Acer freemanii? Because this the species here was not a species that was around until like the early 1900s, like the 1930s. So this person, Freeman, who is a plant breeder, I think this is named for him because he found this, or maybe it was named because maybe in honor of him. He was like the director of some arboretum. I'm a little fuzzy on the details in the history. But anyway, somebody noticed that a red maple, Acer rubrum, and a silver maple, Acer saccharinum, had crossed and developed this tree that had the color and foliage of a red maple, but a lot of the other traits of a silver maple, and hence we have the Freeman's maple or Acer freemanii. So when we have these kind of we have these crosses that happen, what you'll see is a little X between the two, and that means that this is a hybrid. So this is a hybrid between Acer rubrum and Acer saccharinum. Still got the same genus Acer, and with this new species named freemanii, that means that a lot of times when you like take seeds off of this hybrid, they may not come true to type. They may actually end up having way more rubrum characteristics or way more saccharinum characteristics. So the seeds of an Acer freemanii are probably not going to be very reliable, at least like the first few, generations of seeds that you get off of these guys. And they're always going to have this X in the middle there. So that means it's a hybrid. Sometimes you also don't have this species name at all. It'll just be like Penstemon X. And that just means that they're not listing the species name, or there isn't one because it's like divulged from like so many different parent plants and it was never given a species name. That's actually how a lot of like patented cultivars are named. They don't want to give you any information about like the parent plants because they don't want to tell you all of the the breeding that went on to get to this specific Penstemon Prairie Dusk cultivar. There's a bunch of different parent plants that go into that cultivar, but they're not going to try to tell you that because that would be leading you to this patented information that they're making money off of, right? It's probably a bad example, but here's another example here. So this is a Chitalpa. We have one of these in our backyard. I learned some crazy things about them like recently. This is a hybrid between the desert willow or Chelopsis linearis, which note here, a desert willow is not a true willow. So true willows like the weeping willow and that sort of thing are in the genus Salix. Desert willow, not a Salix. So it's in this genus Chelopsis. So it's pretty distantly related when you have different genera, right? And it was crossed with the southern catalpa. So the southern catalpa or Catalpa bignonioides is this really tall tree here. You see this guy down here. It's a very, very tall tree native to the southeast. And our desert willow is a shrubby plant native to the southwest. And this was actually bred by a Ukrainian person back in the either like late 1800s, early 1900s. And they crossed those two together. And eventually, when that plant was brought back to America, they renamed the genus entirely. So this is something you can do when you create new hybrids, is just rename the genus. So it doesn't have Catalpa as the genus, doesn't have Chelopsis as the genus. They've smashed them together. So it's Chitalpa. Tashcatensis is the the species name, and we've got this X here to indicate that it's a hybrid. Here's an interesting thing that I learned about the Chitalpa, and this is wild. So I told you earlier that if you save seeds off of a hybrid, it's less likely to be true to type, right? So how are you going to propagate hybrids? Like, how are Chitalpa propagated? They're propagated by cuttings. So you would take a cutting, this is a perfect place to take a little cutting right down here at the base, take that cutting, pop it into some soil, right? And then you have a new Chitalpa tree. That's clonal propagation, right? It's vegetative propagation. So when you're doing that, you're taking the exact same traits from the parent plant, passing it on to that next clone, right? As it turns out, this Chitalpa that was developed in Ukraine, at some point, folks realized, like way back when it was developed and propagated, like that mother plant had a bacterial infection called leaf scorch, which is caused by the bacterium Xylella fastidiosa. And now every Chitalpa tree that they've tested for this bacteria turns out positive for it, because it's a clone taken from the mother plant who had this disease, and now all these other clones also have this disease. It is mind-blowing and crazy to me, because it's actually the disease is kind of a big deal, especially for other plants, not so much the Chitalpa. So yeah, I don't know, it's a crazy thing that I learned. Wanted to share that with you guys. And I hope you've enjoyed this lecture. If you have any questions at all, please let me know. I will, if I get enough questions, I will post answers in an announcement, just in case other people have the same question. So please don't be afraid to reach out. And I hope you have a wonderful week. And I will see you all again next week.